truly a turning point, I think.
Do you think this is a culture we should bring to Germany?
You have to have natural curiosity.
Hello everyone! Today we welcome Professor Baumeister on set, and today we've curated a series of photographs in which Professor Baumeister will be guiding us through and also telling his story through all of them. Can you please introduce yourself to the audience first?
Yeah, my name is Wolfgang Baumeister. I'm at the Max Planck Institute of Biochemistry in Munich, Germany. But since 3 years now, since the end of the pandemic, I also have a lab in Shanghai at Shanghai Tech University.
Perhaps we can now start. So here we have prepared a series of photographs. Allow me to first share with the audience. So here you can see we do quite have a few of them. So now what's going to happen is I'm going to ask Professor to just randomly pick one.
Well, that is the cover of a booklet which I wrote in my last year in high school. I think I had been given a project by a motivating biology teacher in plant ecology. So that ran for something like 3-4 years. And basically it was, the development of plant societies, after deforestation. So I think it was basically a project I had to do without basically experimental tools, just observation and statistical analysis of some kind. A little booklet, and I used it to compete for the German science fair. And I think that was quite successful. I won a couple of awards for the project in my last year or two in high school.
That's amazing that you made this all in high school. Was it then that you kind of realized that you wanted to go into science?
No, I wanted to explore. It was for me more like a sort of test, and I think I really enjoyed doing that project. I enjoyed that more than other parts of the curriculum in school. It was something where I could be creative myself, and I think it reinforced my feeling that doing some kind of research would be the thing I would like to do professionally. Okay, that is much more recent. That is from work done with cryo-electron tomography when it was already pretty advanced. The task of my group was basically to see how neurotoxic aggregates involved, let's say, in Huntington's disease or ALS interfere with the cellular environment, how they become toxic. And here what we see here is basically an aggregate called a glycine-alanine aggregate. So that is typical, I think, what is found in the brain of patients suffering from ALS.
Were there any particular challenges that came in the way for the discovery?
A cell is a fairly large entity compared with the field of view of tomography. In tomography we look at an area, let's say, of 200 by 200 nanometers. And the cell, is of course, a lot larger. So, targeting and hitting and preparing, the area where the aggregate is located can be quite challenging, yeah.
Here, let's see for our third photo.
Yeah, and that is relatively easy to explain. That is basically my group at Shanghai Tech University. That is about a year ago, I think. The group is now something like 23, 25 people. That is a photograph which is probably around a little bit more than 30 years ago. I'm on the right-hand side. It was the board of directors of the Max Planck Institute of Biochemistry, and by that time I was by far the youngest director at the institute. When I retired 3-4 years ago, I was of course by far the oldest. Two of my colleagues there are Nobel laureates, Robert Hoover and Butenandt, who got a Nobel Prize during the Second World War. And he was not allowed by the government to travel to Stockholm and to receive the award. So he received the medal, at least after the war, but the prize money was gone. That is the molecular machine we are most interested in. It's the proteasome, I mean a multi-subunit complex of something like 2.5 megadalton and 35 different subunits. So it was quite a challenge to determine the structure of that complex. It's taken us something like 10 years to get to this structure at atomic resolution. I think what you see there is the human 26S proteasome at a resolution of something like 3.8 angstrom. Yeah.
Was there a kind of eureka moment when you finally got to the structure after 10 years, or was it more like a slowly building?
Well, we worked on the whole issue for a long time, and what you see in red in the middle, that is the so-called core complex. That we had solved quite a bit earlier by X-ray crystallography. That we solved in 1995. At that time, it was not yet generally accepted that the core complex, is basically dependent on regulatory particles. These are the particles at the ends here, in order to become functional. So I think there was a lot of discussion about that. Quite a complex and a very dynamic molecular machine, so it changes its conformation during the functional cycle. So it's not one structure, so it had to be something like 10 structures that had to be solved. So it took quite a while, yeah.
So it was like getting to each of the structures that took the longest time, right?
Yeah. Okay. That is something relatively early in the development of cryo-ET, that is herpes virus. So in the beginning, when the method was basically working, we could only apply it to relatively small objects, because they were small enough to be fully electron transparent. The eukaryotic cell is by far too thick, so it's not transparent to the electron beam. So particles like, virus particles and bacteria or archaebacteria, that was what we were able to look at in the early days of cryo-electron tomography.
We only have 5 more photographs left.
Yeah, that is a photograph of my predecessor as director at the Max Planck Institute of Biochemistry. His name is Walter Hoppe. He was by training a physical chemist, and he was, I think, a pioneer of X-ray crystallography in Germany. But later in his career, at the time I moved to Martinsried, he became convinced that there are too many restrictions in X-ray crystallography, that I think electron microscopy, which was not yet cryo-electron microscopy, that came a bit later, would be, I think, have more opportunities for the life sciences. He was not thinking about cells. He did not like cells. He was interested in molecules, and he wanted to move there from X-ray crystallography to single particle electron microscopy. Well, that is from my time as a student, where I did also my diploma thesis, or the equivalent of a master thesis, that is the University of Bonn. What you see in the foreground, this building there, was the chemistry building. So I think they had no biochemistry courses at the time, but we had to do standard organic chemistry there. I think I have still mixed feelings about the place. The place was terribly contaminated with mercury. I think if you looked at the floor, and I think everywhere where there was a little gap, you saw the mercury. The building was taken down shortly after I left Bonn.
So no more mercury?
Yeah.
We have 3 photographs left. Let's see.
That is just another photograph of my group at Shanghai Tech University. So I think once a year, I think they are keen to take photos of the group. I think that is pretty uncommon in Germany, but I think it is will be common in China.
Do you think this is a culture we should bring to Germany?
Yeah, maybe, yeah.
Okay, 2 more photos left.
That is not related directly to me. This is an electron microscope as it was used, I would say, in the 1960s and 1970s. You see 4 gentlemen around that microscope. On the left-hand side is Helmut Ruska, who was my PhD supervisor. He was a pioneer, an early pioneer in electron microscopy. He was the first to visualize virus particles in the late 1930s. On the right-hand side is his brother, Ernst Ruska. Ernst Ruska is the inventor of the electron microscope. He built the first commercial electron microscope with the Siemens company back in 1936. Even though he was a true pioneer, it took 50 years until he was recognized for that with the Nobel Prize in Physics. That was 1986.
And I heard your journey in microscopy started because you found an abandoned electron microscope.
I mean, studying biology at the time was very boring, I have to say. It was basically, we were dissecting rats and mice or birds or whatever, and then we were expected to do very nice drawings of what we saw. We got a very good degree if we were very good in making drawings of that. One day I detected an abandoned electron microscope in the Institute of Agricultural Botany, and that attracted my attention. And so I somehow managed to put that into operation. I taught myself the art of specimen preparation. That was my master's or diploma thesis. I was doing that as a kind of an amateur, and I felt then I should try to get some more professional training. That is why I switched then to the University of Düsseldorf to join the lab of Helmut Ruska.
Now we have one last photograph.
Well, I think a kind of a milestone in a way, even though it looks a little bit ugly on this one here. 2002, as a matter of fact, it was the first successful application of cryo-electron tomography to a eukaryotic cell. I mean, we had learned to grow cells directly on EM grids. And what you see here is basically the actin skeleton at the periphery. It was still before we could do proper thinning by FIB milling. So it is basically the periphery which was thin enough to be electron transparent. But it sort of was game-changing, if you like, in the sense that it was selected by the journal Science as breakthrough of the year. 2000. So I think they began to recognize there is a lot of potential. And from then on, I think people began, I would say, to take cryo-electron tomography serious and to see that it could become useful, even though by today's standard it looks kind of miserable.
I mean, today's standard only existed because this happened.
Yeah.
Would you say this is like the work where you feel the most pride?
I think we were very excited at the time. It was recognized, at least some people recognized that this is a kind of a breakthrough. It was truly a turning point, I think, in the development of the field.
That's amazing. The field would not be where it is today without this. So that's all the photographs. And if you would have any tips or any insights that you would just like to give to the next generation of scientists or still a student trying to venture into science, is there anything you would like to say?
I think there is no recipe to become successful. I think what is important is, I think you have to have natural curiosity. Then you can also become passionate about something. And that is important because, I mean, whatever you do, I think not everything will be a success. There will be failures, setbacks many times. And then I think if you are truly passionate, then I think you will develop the persistence to continue.
That's beautiful. So I think this is the end of today's video, and thank you very much for joining us today.
Yep.
Thank you, Professor.